Okay, so hearing not quite as easy as you might think, and neither is equilibrium. These are certainly two of the hardest parts of the special senses to get down. I strongly recommend looking in the face-to-face PowerPoints for maybe some extra little help. There's some videos in there that may paint a better picture than I'm capable of doing right at this point, given the medium that we're trying to communicate by. So equilibrium's in the control of your movement in three-dimensional space, and it requires the vestibular apparatus. This is composed of receptors for equilibrium, and there are several different receptors actually in the inner ear. There are three semicircular ducts that have particular patches for this, patches of hair cells, as you might guess. And then there are two chambers that are going to be responsible for helping us, for assisting us with equilibrium. And they're called the saccule and the utricle. And one is in the interior portion, one is in the posterior portion of the area of the vestibular apparatus. So what is the difference between these two sets of receptors? The three semicircular ducts are primarily there for angular acceleration. So that means like turning a corner in a car, if you're a gymnast, when you're flipping and so forth. So rotational acceleration of the head. Whereas the saccule and utricle are responsible for linear acceleration. Okay, so equilibrium. First we'll talk about static equilibrium. This is when the head and the body are stationary, at least in perception-wise. Dynamic equilibrium is when there's a notion of motion, if you will. Okay? So linear acceleration is the change of a velocity in the straight line. And for dynamic equilibrium, the angular acceleration is when you note rotation. Okay, so those are the two forms of acceleration, and that's a form of dynamic equilibrium. With static equilibrium, we're talking about the body and the head being perceived as motionless. So this is going to involve the saccule and utricle that are part of what's called the macula. Okay, the macula is this patch of cells, and we see a representative here, representative sample of this patch of cells. We see the vestibular nerve. Here are our hair cells that are projecting up into this cup, into this material that is very gel-like, just like what we saw with hearing. On top will be these stone-like components called otoliths that will help weigh down this gel-like material. And so you should know that the saccule lies vertically, okay, whereas the utricle lies horizontally. So we have two patches of tissue that are going to adjust with our head position and are going to tell us what position our head is in relative to the body. So here you go. You've only got about 40 to 70 of these cilia that are projecting off each hair cell. But there is one true cilium. It's a longer one. You can barely see it up there sticking up. It's a little longer. And it's embedded in this membrane. All right, so the membrane, again, is weighted down by these otoliths, which are made up of calcium carbonate that add weight and inertia to the movement of this gel-like material. So if we have our gymnast standing straight up on the balance beam here, then we see exactly what we'd expect in this particular structure. But if she flips now and changes her head orientation at basically a 90-degree angle, now we see that these stones are going to weight down this gel-like material and drag it in this direction. And when this thing sags in this direction, when this membrane gelatinous material sags, it's going to yank on these hair cells, causing them to release or open channels, causing potentially the eventual release of neurotransmitter. So in static equilibrium, in the head tilts, we're going to see the membrane move in one direction for either the utricle or the saccule. Okay. Now, linear acceleration can be detected by these as well, and this will occur when the otoliths are lagging behind the driving force if you're moving in a straight line. And so we'll see this. Let's look at this here. And so what you have is, if you're tilting your head, you see how the otoliths drag the gelatinous material, and this gets pushed in one direction, and that will either increase receptor output or decrease it. So if this particular movement happens to decrease it, then if you did the head movement where it was down and the otoliths dragged the other way, then that would decrease the amount. So the brain can tell which way is the head moving by how many action potentials are being generated in these structures. Now, in the semicircular ducts, we have 3 little islands of cells. They're going to be found in the ampullae. So there's a swelling, and for this third one, maybe that's the swelling right there. And that's what we're looking for. So we're looking for rotational movements to alter the positions of little gelatinous material compilations surrounding hair cells again. And this is going to happen in the ampulla. So let's take a look at that. Here's our ampulla. This is that little swelling, and you see the hair cells here found right here. There's fluid that moves around this gelatinous cup, and if the fluid is moving in one direction, it'll cause that gelatinous cup to move, and it will drag the hair cells with it. And then if the fluid reverses direction, it'll move in the other direction and cause that. And once again, one direction will lead to increased amounts of neurotransmitter, more action potentials. The other direction will lead to less amounts of neurotransmitters and less action potentials. So that's the ampulla, okay, all due to the similar circumstances we saw with the cilia in the process of hearing. So if she's spinning around, rotating, then we'll see that the endolymph will lag behind the rotational speed, and if you're turning your head in this way, the endolymph will move in the opposite way, right? And that will lead to more or less action potentials, but it will stimulate the hair cells. Something will occur, either more or fewer action potentials, and you'll get the brain recognizing that. So here are our projection pathways. They go into the vestibular nuclei and then project onto an area that's going to help us correlate this activity in the vestibular apparatus with that in the eyes. We're going to, of course, connect that to our motor coordination area where we're getting inputs from our muscles and our joints and so forth. And then on up into cortical regions where we process equilibrium information in and around the parietal lobe. And this will help us assimilate where exactly we are and where we're moving to in particular. Okay. So all of these structures will have synapses so that the vestibular part of the vestibulocochlear nerve is formed. They end up on some nuclei in the pons and medulla. And so I don't want to get particularly incredibly detailed with this, but there are 5 target areas for this information that we just addressed, the cerebellum, so that we can put the picture together about how our muscles are acting and where we need to end up at. We're going to have the nuclei of the different eye muscles so that we can keep vision in context with our balance information. And the reticular formation can work then to adjust different autonomic aspects in terms of blood pressure, heart rate, and so forth that accommodate the particular posture changes you may be engaging in. The spinal cord has the vestibulospinal tracts that will deliver the information necessary to muscles that need to act in accordance with the vestibular information. And then finally to the thalamus so it can deliver the information to the correct location in the cortex for proper orientation.